Okay, we're recording.
Okay, hello. My name is Wendy Santana, and I'm a brain cancer survivor. A few years ago, I created a blog called Second Chance to Live, and the purpose of my blog is to share resources and information. Today, I have the honor of interviewing Josh Neman, Ph.D., and we are recording our session. This is a brand new approach for me, and it will be a way of completing the third post in a series I have been working on called From Attendee to Speaker, a look at an ABTA Regional Educational Meeting. ABTA is the acronym for American Brain Tumor Association. Josh is a USC neuroscientist and professor who also chairs various USC departments. Last month, he chaired the ABTA meeting that I have been writing about. He is one of the unsung heroes in the brain tumor community, as the work he does may very well lead to a cure for brain cancer someday. Welcome, Josh.
Hi, Wendy. Thanks for having me.
And did I say your last name correctly?
Yeah, you said it perfectly.
Okay. So let me go to my interview, and I'll just jump right in by asking you the first question. Obviously, I'm a little bit nervous since you're the first neuroscientist I've had the pleasure of interviewing, and because we're recording this. But I reached out previously to both Nikki Kozak from post 1 of 3 of this series and the ABTA's program manager, Emily Lippert, from post 2 of 3 for their thoughts. So the questions are a combination of what the three of us came up with. First off, we would all really like to know, at what point in your life did you decide you would become a scientist researcher?
Yeah, that's a really good question. So my mom is a scientist.
Oh.
Yeah, and so when we were, I was born in Iran and we fled because of political reasons, and she was a scientist and she's a zoologist. And so I've always been in that culture. She studied the migrational patterns of butterflies as it relates to pollination, because its agriculture is really big. And so I remember from very early on, 5, 6 years old, being in her lab. And I think that, osmosis through that experience, I, love science. So it's in my genes, per se. At what point did I decide to become a neuroscientist? Well, I've always been fascinated with the brain. It is truly within the body, the final frontier. And I know that's, we always say that, but it's the truth. It's the hardest, I think, organ to study. And we are just tapping the surface, no pun intended, of it.
Right.
And that's what I studied in college and then my doctorate. And I studied how the brain develops for my PhD. From stem cells. And then I decided to go, well, what happens when things go wrong in the brain? And that's how I got into my brain tumor studies.
Wow. So at what point, though, did you decide that you wanted to become specifically a neuroscientist?
Yeah, I think it's really probably, if I have to think about it and pinpoint something with accuracy, it might have been sometime when I was in 9th grade in high school, when we first started doing dissections in biology classes. And I don't know, it was just something that just drew me to it. So I don't have, unfortunately, like a lot of other things which I need a concrete answer being a scientist. With this, I never have a concrete answer, because I think that's what passion is, right? You just know inherently what you're attracted to.
And you're attracted to the brain.
100%.
The human brain.
Yeah, the brain in general, yes.
So what was your educational trajectory, and yeah, how does one become a neuroscientist?
Yeah, that's a good question. So you start off, there's multiple levels, right? So there's now you can get a bachelor's degree in college. So for all of those young folks who are listening, they could start off, at a university and major in neuroscience. And that's where you just learn very broadly what the brain is, functions. And then I decided to go and get my PhD in neurobiology. And I did some research in between to just solidify my experience and the want and the desire to go into this, because it is more than a job, right? It's a difference between a job and a career and a lifestyle, being a scientist. And I decided to go get my PhD in neurobiology. And I was very interested at that point, which was in the late 90s, early 2000s, of stem cell biology, because that's when it was a very hot, quote unquote, topic. And we were learning about learning and memory, how stem cells, work in the body. And then we found stem cells in the brain. I mean, I didn't, but the field found stem cells in the brain. And that just opened up a new avenue. And that's when I started to study the development of stem cells into a specific type of a brain cell called oligodendrocytes. So it's the cells in the brain that make the myelin sheath. So what that really means, the brain, if you think about the brain, it's just millions of wires, okay, our brain cells, our neurons. And just like the electrical wires we have that we plug in home, those wires are insulated so that you can conduct the electricity. For those listening and going to be reading this, myelin is the insulation, is the fatty insulation around the wires of the brain. And the cells that make myelin are called oligodendrocytes. And so I studied those in the context of making how do we make stem cells into oligodendrocytes? Because patients who have MS, various neurological diseases, those cells are affected. And so that's what my PhD was on in that context. And then after I finished that in 2009, I really decided to go into cancer because I felt like I knew up to that point everything about normal development. And I wanted to see what would happen when things go bad, go wrong. And in order to learn what goes wrong, you have to learn what has gone right at the beginning, so you can detect what has gone wrong. And so it was a natural progression for me to go and study brain tumors and brain cancers.
And I just happen to have an oligodendroglioma, which is from oligodendrocytes.
Sites. I know that. I knew that. The team, you and I, it was destined for you and I to talk.
Yeah. But now that you've brought up stem cells, I know that the basic person like me hears about some stem cells in the news from time to time. And also, I used to work for an agency that supported blind people, and there was a lot of talk about stem cells for that. Can you? I'm not grasping the significance of how stem cells relate to the. I'm not really grasping what you're saying about stem cells and the brain. And so I think most likely others may not understand that connection.
That's a good point. So we, before I would say the early to mid 1990s, we thought that when we were born with all of our brain cells. And I'll just say brain cells collectively, okay, although there are many types. And we thought that we were born with all of them. But what we found out is that within the brain, we have centers that make brain stem cells. And you may think, well, why does the brain need to make newer brain cells? And one of the basic things is that we make new memories. And in order to make new memories, you need new cells and new connections. And so the brain has this reservoir system, early on, that it starts, as we're, younger, and then our teenagers, and even, in our 40s and 50s, it has a reservoir of cells that it keeps dividing, a pool. And those are the stem cells within the brain that are made and then sent out to various regions. So for example, for learning and memory, for smells, okay? And new brain cells are made for new memories and new stem cell and new smells, for example. The other pool, and that's why with respect to oligodendrocytes, it's important. Oligodendrocytes, they somewhat have this, what we call a progenitor pool. What a progenitor is sort of like a teenage cell, okay? So you have an adult cell and you have a progenitor cell, and progenitors are your teenagers, okay? And those teenagers have sometimes they churn over, okay? And with respect to a disease like multiple sclerosis, where oligodendrocytes are affected, the pool of progenitors, so the pool of teenagers comes up after the adult oligodendrocytes are harmed, okay? And when the pool comes up, the brain also shifts where it needs to replenish that pool, okay? So the embryonic, okay, or the baby stem cells. So you have baby stem cells, teenagers, and then you have adults, sort of like natural humans. And so it's like a domino effect. When the adults are gone, the teenagers become the adults, and the brain basically sends baby stem cells and makes more progenitors or teenagers. It's sort of like a cascade. It's natural evolution, if you want to per se think about it, or development. And so that's what I studied, and that's why stem cells are important within the brain. And we are hoping that stem cells can be used, and there have been studies made that they can be used to not only basically develop therapy so that we can inject them, hopefully. And there have been many studies have been done about how stem cells could replace the damaged brain cells. But there have also been studies done that we can utilize stem cells for brain tumor patients to deliver therapies, for example.
So I know that neuroplasticity is a huge thing because, like, for instance, after my craniotomy, I could not move my right side, which a lot of people, and I couldn't speak. And so when I was working with physical therapists and occupational and speech therapists, they would talk about the neuroplasticity. Does that have anything to do with the stem cells? Like,
Yeah.
were my stem cells like recreating themselves, or
Exactly.
I'm
Yeah. So if you think about plasticity, all that means is, the more, for example, you and I talk, the closer we become in our connection, our friendship. Okay, that is plasticity. Okay, and that's how neurons they work. The more neurons they communicate with each other, the closer they become, the more connected they become, and that means neuronal plasticity. Okay, it's sort of like friendships. Now, how does plasticity and stem cells come into effect? Well, when someone, whether they have a brain tumor or they've had some kind of a neurological disease, those connections are sometimes they're interrupted or they're taken away, right? And they need time for it to come back. And sometimes they do come back depending on the disease, and sometimes unfortunately they don't come back. Now, how do stem cells come into this? Well, if a patient has lost their neurons, let's say, to a disease, for example, traumatic brain injury, a stroke, okay, sometimes brain tumors, right? Because they've had that section of their brain taken out, right? So they've done a craniotomy, right? So now that section of the brain is missing, right? The hope for the future, we're not there yet, is how can we, utilize stem cells to make new brain cells so that we can use those to reconnect for what it's been lost. That is the ultimate hope of neuroscience when it comes across, diseases, basically from cancer, brain tumors, first fixing, for us to fix, and find the solution for brain tumors, but also then replacing, right? Because connections have been lost.
So just to clarify, I just, are the stem cells regenerating themselves, or are you researching like putting stem cells in someone's brain?
That's a great question. So both. Okay, so early on, okay, stem cells, early on, what I mean by early on is in age-wise for us, okay, people. The younger we are, the better our brains are able to
Right.
regenerate. Okay? The older a person gets, those stem cells sort of, they become quiescent, or what we call quiescent, or they basically quiet down. So they lose those stem cell properties. So
Right.
if in a person who is in their 70s, 60s, 70s, those stem cells are not as active. So we have to come in from the outside and put them back in.
Oh, that is so cool.
So it's a balance,
um,
right?
okay, moving on to the next question. Um, you received a, a grant from the ABTA. Can you please talk about that and what, because the ABTA gives out research grants for people who don't know. I think it's pretty competitive. And can you talk about the research grant that you received?
Sure. So yeah, I love the ABTA, not because of the things, because of the people it allows me to interact like yourself, but they are super huge supporters of scientists like myself. And so my initial laboratory studies started with brain metastasis. So cancers that spread to the brain from the outside, like breast cancer.
Right.
And because of those research studies, we found certain molecules that these breast cancer cells were using to survive within the brain. And then I came and asked the question, well, this certain molecule, because I am a neuroscientist, I knew that it's expressed in one part of the brain very uh, uh, strongly. It's the cerebellum, which is the back of the brain, which helps us move, coordinate movement. And I said, well, this molecule, we found that breast cancers use it. I know that the cerebellum also has this type of this protein. And I said, well, what can tumors that come from the cerebellum, the back of the brain, use the same molecule and pathway? And one of the main cells in the brain, in the back of the cerebellum, that becomes cancerous is this childhood brain tumor called medulloblastoma.
Right.
And medulloblastoma, we found out our initial studies really uses this other molecule called ABAT. Okay, so it's funny. It's sort of like the same acronym as ABTA, but it's
Right.
ABAT. Yeah, I don't want to confuse your viewership, but that's what it's called. It's just called ABAT, just for learning purposes. We found out that in breast cancer cells, ABAT is used for brain metastasis. The cerebellum has a lot of ABAT, and the question was, well, then a tumor of the cerebellum, can that use ABAT a lot? And that's where medulloblastoma comes. And that's the funding that I applied for the first time as a medulloblastoma researcher. And the ABTA really, it's a very competitive granting program where we have basically, you apply nationally and internationally. And I was very fortunate enough that, the review committee believed that even though I hadn't done medulloblastoma research, I could bring in a new insight because of my other studies. And I received this grant, and they were the first foundation and grant agency to give me this. And we are actually in the process of publishing our data. And we found out that this protein, this AB, this ABAT is basically, it's the cause of these tumor cells, these pediatric tumor cells, getting up and leaving and going into the CSF. They are one of the main roots of the problem.
And for those who don't know, can you explain what CSF stands for?
Yes. So I'm sorry. So CSF stands for cerebral spinal fluid. So imagine the brain, okay, and our brain is sitting in a bath, okay, in this fluid system for protection, for nourishment. But unfortunately, in a lot of cancers of, brain tumors, tumors spread via this fluid. And the reason is because we have these canal systems within our brains called ventricles, sort of like the Venice canals. And so tumor cells can drop from the tissue into these canal systems and now travel all over the brain. And that is called leptomeningeal disease. And so we found that this protein, ABAT, is important in those cells that want to get up from the cerebellar tumors, the medulloblastoma tumors, and swim around the CSF. And this all would not have, none of this would have been possible if we didn't get the first seed grant from ABTA.
The ABTA rocks.
Oh, 100%.
I love the ABTA too. So, and were you done with your response before I rudely cut you off?
Oh yeah, that's it. No, that's it.
Yeah, that was deep. So what motivates you to research brain tumors and why focus on brain metastasis? I think you answered this a little previously, but can you add to that?
Yeah. So I think, besides the brain being the final, frontier, unfortunately, cancer hits everybody. Okay. It's not, cancer is not biased. Everybody in their family has someone who is, who has had it or who has unfortunately succumbed to it. And when I first started, studying brain tumors, it was because of scientific intrigue. But then within my first year, my aunt got brain metastasis.
Oh.
And so I was studying the disease, that my aunt had. And then it became more than just a profession and a scientific, journey. It became also a very personal one. And so, since then, I've had, unfortunately, many, I've had friends who've passed away from it. And so that, while I have the scientific passion and drive, I also have the personal drive to it. Yeah.
Wow. And just curious, is the aunt your mom's sister?
So this is the thing with Persian, it's my uncle's wife.
Okay.
Yeah, my mom's brother's wife, but I call her an aunt because we were very close.
So speaking about brain metastasis, I can't say certain words, but
Okay.
Nikki Kozak, who was in the conference, and I wrote the first blog about her. She mentioned that she is seen by an oncologist who specialized in melanoma patients. She said he was able to grow and test a sample of her specific tumor cells in his lab and found a very unique mutation, which led to a research project that opened up a new treatment door for her. And when things were really going downhill fast, it sounds like she's been through just hell. And so she continued that the success of the treatment turned out to be vitally life-extending, getting through FDA approval of Keytruda. Is this an example of something that you and your team were involved with? Please explain.
Yeah, so I wasn't involved with her, but we do this, the whole goal of personalized medicine, okay, and what she went through is a perfect example of personalized medicine. Is for a patient to come in and they, the surgeons take out her tumor, okay, they send it into the research labs and we work on it, and then we figure out what is wrong specifically, what kind of mutations, exactly what you described. Okay, and we find out what mutations are, and then we can basically come up with a personalized therapy for that patient. Okay, while I was not involved with hers, we get these we get these patient samples back, from the OR, from neurosurgery. So my
Right.
primary appointment is in neurosurgery, and my colleagues, we get tissue and we work on them. We're not. My hope is that we get there, and I think we will within the next year or so, where it's exactly what how you describe for her. We do it in the lab here, and this is honestly this alludes into why we need so much support and funding for research.
Right.
Okay, because it takes a lot of manpower. It takes infrastructure to do this, and that is our goal, right? Rather than, my goal is, this, while the science is amazing, we want to give back to the patients. Okay, and we need the support to build those infrastructures in place, so that we can have a patient's tumor come in from the OR, get into here, do a fast turnaround, or a sufficient turnaround of us studying that tumor and finding out how we can tackle it, with new drugs, new, what we call, modalities that are not there yet.
I was just thinking about how she's so clever when she goes into USC. Uh, I don't know if you saw it in my second post. Uh, I mean, in the post I wrote about her, she created a moonshine that she,
Yeah.
but recently, like last week, I think she went back there for a full day of testing. It's not a day at the spa.
Uh-huh.
And um, she actually made chocolate covered walnuts and put pecans on them for brain metastasis.
Yeah. So that's
She's
the,
so clever.
but it's also, it's that mentality of not allowing the tumor to basically take over you, literally, right? Psychologically, right? And it holds true. I mean, I've had those experiences, where we all have, none of us are superhumans where none of this affects you. So I think that's a great mentality, and I think we should all, when life literally gives you lemons, you make lemonades out of it.
Yeah,
Or
that's
in her
what.
case, walnut cookies.
um, so where, I think you've also alluded to this, but where do you find your inspiration and ideas for your research?
Honestly, I did the things I said, but I think, a big part of my inspiration also comes from patients. When I speak to patients, I hear not just their story, but I hear, for example, the things that they're lacking. So you talk about plasticity, and I'll use this as one example. About 2-3 years ago, I was talking to, patients, breast cancer patients, who I bring into my lab. And by the way, I know I've told you this before, but when the pandemic is over, you have an open, invitation to come into the lab.
Oh, thank you.
But I basically, I get those inspirations and the stories from them. And I'll give you this one example. A couple of years ago, had breast cancer patients come into the lab, patient advocates like yourself. And they were describing, because they knew that my background is neuroscience, and they said, when we were getting chemo, we felt like this chemo fog and this, brain fog. And why is that? Why? And I started describing to them, because there is research done on chemo brain. But then something sparked into, in me of, well, what if, this chemo fog and this chemo brain contributes to their cancer and cancer spread. And so we started working on those. So, those studies. And so my point to you is that I get inspiration from talking to brain, brain tumor patients as well.
That's why you're the, I, 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 I'm, I'm assuming that many scientists look purely at the science, but what makes you different is that you're interested in the people behind the cells.
100%. And we, and I tell my students in the lab and the postdocs in the lab, I have patients, coming through the lab all the time because, as scientists, unfortunately, I mean, it's great that we are focused, but I want them to remember why they are coming into the lab every day.
Nice. Yeah.
What is the bigger picture in all this? That yes, it is for the scientific venture and the exploration, but ultimately it's for for the patients. And I always say, and I don't say it as a joke, I say, I hope one day I'm out of a job.
Yes, I hope so too.
Yeah.
But that's just beautiful. Can you please share, going back to the ABTA conference, I don't know how you have time for all of this, but can you please share a little bit about your role as the chair of the ABTA's Regional Patient and Family Meeting? I know you chair several things for ABTA, but I'm writing just about that most recent meeting. And specifically as the chair, how do you develop the program and what informs you to select certain topics and speakers?
Yeah. So, I think, first of all, I always have time for, patients and doing these types of meetings, especially, the regional meetings with ABTA. What really, it's working with Emily, Leeper, she's amazing, and the
Yes.
other members of ABTA staff. The way, the program, comes to fruition is we have several meetings months before, and we go back and forth. And I basically, and it goes back to, talking with patients throughout the years, throughout that year. And you sort of pick up the nuances and the things that patients want to learn, because ultimately this meeting is for you guys. It's not for me, it's for you guys. And I remember topics, or if I'm talking with patients, and they say a key word that sort of sticks in the back of my brain, and I write it in my, in my phone notepad. And then when it comes time to, putting these meetings together, I think, I go back to those conversations, and I say, okay, we need to have a topic on, for example, we had a topic, on stem cells, cancer stem cells. Harley Kornblum from UCLA gave it. And then, or immunotherapy, and so these are, it comes from speaking to patients. Okay, that's where I get the ideas from. So it's, so it's all because of you guys, and and the meeting is for you guys.
Oh gosh. Okay, so I rewatched your session on brain metastasis during the portion of the meeting on how new breakthroughs are discovered. And I'll say it again, the way you put the presentation together was phenomenal, and a link will be included to that presentation. I just really think you have a gift for breaking down a highly complex topic that very few people can understand, and you make it relatable. So I sent you a few screenshots that represent a few of my favorite slides from your presentation, which you also had animated slides that on top of that were great. But I would like you to pull up the screenshots now.
Sure. Let me pull up my screen. Okay, you see my screen?
Yes. So the first ones, yeah. So you, and as you know, since my brain tumor was located on my left frontal lobe, it affected my speech. And
okay, take your time.
Yeah. You started off talking about the origins of cancer and how it is more complex than we think. And then you went to engaging analogies. The next slide.
Right. So I'm going to put this into PowerPoint mode so you can see it. You still see it?
Yes.
Okay. Let me put next slide. Go ahead.
Yes, that one.
Uh-huh.
And you made analogies about the complexity of the tumor microenvironment necessary for brain cancer to grow. You compared, excuse me.
It's okay.
You compared brain cancer to wildfires, which at the time there were actually wildfires going on the West Coast, stating that similar to wildfires, all the right conditions such as drought, wind, weather, temperature, etc., need to be present for brain cancer to develop. Can you please reiterate what conditions are needed for brain tumors and cancer to grow?
Sure. So what the term, for those members of the audience, I'm going to go back one slide and come back to this. Okay, very fast. So what ends up happening is that we had this notion, and can you see my cursor?
Yes.
Okay, good. So we had this notion, an idea, which is true that, tumor, which you have here, cancer, just is abnormal cell growth. Something happens in the middle here, and these cells go bad, and then all of a sudden they start growing. And then you said it very beautifully, the analogy that I gave to wildfires, because it's not just the growth of these cells, but what cancer does, and cancer is very smart. Cancer recruits our normal cells in the body to come and help it. Okay. And this is that tumor microenvironment that you alluded to. So if the cancer cells are here in the red, the things that are necessary for, the microenvironment is a city, it's an infrastructure. Just think about that. And I know I have a picture of an infrastructure, a metropolitan in my next slide. It's this, and I'll go back. So it's this metropolitan. So as I'm talking, just imagine this. So I'm going to go back to this now.
Yes.
This metropolitan, you have the cancer cells, but the cancer cells also come in and tell other normal brain cells in the cancer, in the instance of brain cancer, but they also tell our immune system, for example, to stop looking for me. Okay, I am a normal cell, because what our immune system does normally is it fights off cells that are not normal, whether it's from a virus, a bacteria. Those aren't cells, but to bad cells. We have bad cells in us all the time, but our immune system catches those errors.
Right.
Okay, our body is, imagine if you're typing something in Word, you're going to get spelling errors all the time, but there's a spell check system. And that system is the immune system. And part of the immune system, cancer cells, what they do is they trick the immune system. They say, hey, don't come and get us. We are actually the normal cells. So they're sort of like, that analogy that I always use, the wolf in sheep's clothing.
Yes.
They mask themselves. So that's one facet of it. Then the other facet is these brain cells, brain tumors need nourishment. So they need this highway here, blood vessels. So they hijack the cells that make the blood vessels. These are called, let's say, these are the endothelial cells, okay, cells that make the blood vessels. And they say, hey, come up and set up a highway system for us so we can get food, we can get nourishments. And so this whole concept, the microenvironment, is a city where the tumor cells initially, they act like they're helpless, and they ask other cells in our brains, okay, even those brain cells. Remember those, you were talking about neuroplasticity.
Yes.
Neuroplasticity involves molecules. Okay, tumor cells, and this is one aspect where my lab is studying, is what kind of molecules are the tumor cells hijacking from our neurons, for example, okay, for nourishment.
Right.
Others, are studying, well, how does the immune system come in and basically gets tricked, okay, into not becoming active. Other people are studying what's called the angiogenesis, or the vascular portion. So how do tumor cells set up these highways in the brain these blood vessels for their own nourishment, and that's called the blood tumor interface or blood tumor barrier. Those are the words. So that is ultimately, it's called the microenvironment.
Whew. Yeah, that's, I don't even, thank you.
Yeah.
Um,
No, and I know it's, listen, this one slide could be one
I know,
semester's worth of courses.
I get that. It's, yeah, but it's a great visual, and you do such a good job of explaining it.
But that's the whole point, for me, as someone, I going back to, we talked about my education. I took a very, important course in my undergraduate, this education course. And my professor back then at UCLA, he said, there's no point for, a professor to stand up here and sound so complex, so he puts himself on a pedestal. That's not the point.
Right.
The point is for us to convey the message and teach. And in order to do that, I hate using big words, when I talk. And it's not because you don't understand. It's sort of like, if you're an accountant, if you want to use accounting terms with me, great, but you better use words that I could understand so that your message gets conveyed. And that's what I try to do, and that's my goal is for everyone to understand the process that what we're studying in the lab. It's not for me to stand up here and sound smart.
Oh, God, that is incredible. Another great example of how you made your presentation interesting for those of us who lack a scientific background was by utilizing slides of famous people such as Chadwick Boseman, Ruth Bader Ginsburg, and Taylor Swift's mom. Can you
Right.
please pull up those slides right now?
Yes.
And share what you would like people to understand about how those slides relate to brain metastasis.
Sure. Before I get to get to those slides, there's this one animation that I'll go through, and the term metastasis it's a misnomer because people think that, and I'll use breast cancer. That's the easiest because a woman will never die of her disease within the breast. So these tumor cells are in green here, okay, and a woman will never die because of these tumor cells here. Okay, and it's when these tumor cells pick up and they travel through those highways of the body, right? So the blood vessels, and they metastasize and they spread. So the misnomer is
That
that
is great.
yeah. So when a woman, any type, this could include skin cancer, for example, melanoma, lung cancer. When we talk about brain metastasis, it's not where the brain is now bad, okay, in the case of metastasis. It's when these cells pick up and now they're growing in somewhere else. So it's breast tissue growing in the brain, okay? And now this is how, and if you think about it, this applies to any type of cancer. So, in Chadwick's case, colon, you can take out the colon, okay, and a person would survive, okay? But what, unfortunately, in his case, it was when those colon cancer cells, they picked up and they left to other parts of the body. And that's what ultimately comes, to the demise of the patient, or in this case, unfortunately, the Chadwicks. The same thing with, Justice Ginsburg. She had pancreatic cancer, when patients who have pancreatic cancer, one of the treatments, I don't know what her treatments were, but I'm using this as an example. They do what's called a Whipple surgery, where they literally chop off, the head of the pancreas where the pancreatic tumor is. Great. They chop off, person can still survive with, the majority of their pancreas, or not having, their pancreas. I mean, they go on to having an alternative, nutritional lifestyle, but you can survive. But what ended up happening in these cases, even with Justice Ginsburg, those tumor cells got up and they left, metastatic. Okay, and when the metastasis happens, that's when it becomes really hard. It's going back to that analogy of the wildfire. It becomes wildfire that it continues to spread very, very fast, and we can't contain it.
Oh, God. So, but why is it? Um, so there's a blood-brain barrier that you referenced earlier.
Right.
And I know that for primary brain tumors, they're hard to treat. If my understanding is correct, because of the blood-brain barrier. So it's how does metastasis happen to the brain? Is that barrier not? I don't know how to frame it.
But you're on the right
Is
path.
that?
Finish your thought.
Uh,
You're on the right path.
uh, it seems like when you have things can cross, an existing cancer can cross that blood-brain barrier easier than trying to get something from the outside in to treat what's already in the brain. And um,
Okay.
um,
You're right. That's exactly the exact statement. So I know we didn't talk about, I'm gonna go to that slide. Let me fast forward about 5 slides for you. So it's this slide. So this is, the primary tumor breast cancer. So let's say whether, the cells, it's exactly what you were saying, is that cells, they travel through the circulation and they get to this, what's blood-brain barrier. And the analogy I use as the blood-brain barrier is the TSA agent at the airport.
yeah.
Okay. Tumor cells are really smart. They've learned a way to tell the cells here, okay, which include, there's about 4 different cell types, okay, and they've learned to communicate with those cells, and they say, hey, let me in. So remember that tumor microenvironment, what I said, they learn to
Yes.
trick the body. They learn to trick these TSA agents to letting them through without a scan, okay, and they
Wow.
grow. But unfortunately, molecules, for example, like chemotherapies, they can't talk, right? Or I mean, not per se, I mean, not literally talk, but those chemotherapies that we try to get up into the brain have a harder time because the TSA agents block it. But tumor cells have learned sort of like the language of the brain. And this goes all back to, communicating with brain cells to trick them to say, hey, let us in. We're okay. And so it's this hijacking that I constantly allude to, and you were 100% right. So these tumor cells can get into the brain easily, but not a lot of therapies. And we're trying to figure out ways of finding drugs where the TSA agent is okay with letting it go through.
Yeah, that's fascinating because I mean, like it's like if maybe perhaps an understanding of how breast cancer travels to the brain could lead to um, a cure for brain tumors by tricking by like putting breast cancer in the brain somehow or something.
Right. Right.
I mean, that is such a layman term
No,
for
but you're
with
right though. That's exactly what it is. You're right.
um, so what are a few of your research goals?
Yeah, but let me stop my. Oh, actually, no, no. You know what? I'm going to use some diagrams to show you what one of my research goals are. Let me go through this. So my research goals, it's 2. It's how do we stop the communication of tumor cells and brain cells? Okay, so in this analogy, and this is actually a microscopic image of something we did in the lab. So the tumor cells are in the red. These are brain cells, okay? And it's that plasticity that you talked about. So that communication, okay? The communication happens right here, okay? Where the hands of the tumor cells meet the hands and the feet of the brain cells. And our goal, my specific laboratory goal, is how do we stop these tumor cells and brain cells from communicating? Not only in the case of metastasis, but in the case of pediatric brain tumors, right? Because if we can stop that communication, then we can isolate that tumor cell and unmask it and basically starve it.
Yeah.
Sort of like suffocating it. So I'm glad that you brought up the analogy of the wildfire, right? It's sort of, how firefighters, they basically dig a back fire line, right? To starve the fire.
Yes.
Okay, they draw those lines. That's what we're trying to do.
Fascinating.
Yeah.
And what do you enjoy most about the work you do?
Yeah. Let me stop my share screen so now you can see me.
You
Let me hold on one second. So you
make such good analogies. It's incredible.
No, but that's the whole thing. Sorry, so your question was, what do I enjoy in the laboratory? What I enjoy doing the most?
Just in general about the work you do. It
Yeah.
could be the laboratory or something else.
No,
The
100%.
teaching,
Yeah,
I don't know.
it's all encompassing, but I'm the kind of person that is never satisfied with not an answer, but I want to know the why behind something. Okay.
Which is probably fundamental in being a scientist.
100%. And it's the why that gives me the energy to wake up, okay, in the mornings, whether it's in a pandemic or not, that drives me into, reading, about discoveries. And it's the curiosity, okay, and that's what I enjoy the most, and that's the satisfaction I get, and so yeah, I think that's the most enjoyable thing and that's the nourishment for my brain.
So what are a few of the challenges that you and your team face?
Yeah, I think we alluded to this. Honestly, I think I'll put it to you this way, actually. A lot of people right now during, the pandemic, the COVID pandemic, we just came out with a vaccine, last week, and you and I talked about it. I think this is what a lot of people are like, well, how can we come up with a vaccine in such a short amount of time? And, how could this be? And the way I look at it is, this is what human ingenuity is that if we all are in it together with this drive and passion, you see how fast we can come up with a solution.
Yes.
That's the beauty of humanity, and I think the big thing is that I wish that we could carry this energy into other diseases.
Yeah.
Where we have this drive to cure brain tumors, to cure metastatic, tumors, to cure all of these other diseases. And we see the beauty of what happens when we act as a team. And while we're not fully there, this is what the support we need. And so whenever I say, you were alluding about personalized medicine, well, we need an infrastructure to be put together. I think the thing that our team needs and science needs is support. Okay, is the fundamental support from folks out there to know that there are people that are fighting, on their behalf to make things better. Okay, and I think and we see what happens when we can put our minds, our thoughts, our funding energies together, because none of this could happen by just, I could sit here and be blue in the face till next year and come up with great ideas, but they're just ideas and we need to test them.
Right.
And unfortunately we don't, in the current climate, it's very hard to test a lot of ideas, not just for me, but just for in research and science in general.
Yeah. So speaking of the pandemic, how has that impacted a typical day in your life? And what is a typical day in your life? You have so many titles at USC. Can you please summarize those and tell us what a typical day in your life is?
It's funny, there is no typical day in my life. That's the cool thing about it, right? Every day is I do something different. Yes, the research is the main focus, but the typical day, it could be from working, with my team, my graduate students, my fellows within the lab, us going, and that's what I enjoy the most, is where I get, as a professor, you sit and you do all these auxiliary things, but my true heart, I'm a scientist, and the days that, that I sit with my team members, and we go through data, and I go on the microscope, but I look at the results, and we brainstorm together the results. That is the most exhilarating, time for me. The other aspects is mentoring, so I chair our cancer biology Ph.D. program here at USC, and I have about 30 Ph.D. students under my belt, not doing research in my lab, but just in our program, which I help, with various facets. And so these are the next generation of cancer fighters, and I want to see them succeed, because if they succeed, that means we succeed.
Yeah.
And then, then I have various administrative roles, helping, other lab, team members within neurosurgery and our new brain tumor center that we just launched, and so we want to see that community grow. And then I do a lot of community outreach, things like yourself. And all of that is, my schedule for, a week, or, but it's never the same. And that's what I love about it.
So lastly, inquiring minds would like to know, like, as Nikki says, what do you do when you're not elbow deep in slides and cells?
I love spending time with my family, like anything else, kids. I love basketball, so, with my son. Just, I think I honestly, I know it sounds very cliche and poetic to say, but I want to leave this world a better place, not only for my family, but for other people, and I think the right balance, when I spend time with them, they also energize me to come back into the lab and to do better for humanity. And I know that sounds cliche, but it's true.
Oh no, my saying is, do as much good as you can, where you can, while you can.
Yeah.
And I'm a humanitarian as well.
Yeah.
So, and I have to say, you are doing so much good. And I thank you very much for your time, Josh. Is there anything else that you would like to share before we bring this to conclusion?
No, I mean, I think you did a great job, actually. I think, I'm always open to having, folks, come through the lab. If anybody has any questions, I'll put the link on. But I put this, it's funny, with the pandemic, you find new opportunities. So if, this website that you see behind me, that's my lab's website. If you need more questions, just go to it. If you want to get, your audience needs to get in touch and, email or set up some time to come through the lab, I really look forward to it.
I'm going to take you up on that.
100%. Good. I'm expecting it then.
Okay. So
that's it.
I think that's it.
Okay, good.
Thank you so much.